Hello students, welcome to Sacred Heart High School. Today we are going to study in Science-2 chapter number 1, Heredity and Evolution part 1. Before we begin with our chapter, we shall study about the famous scientists and their contribution. The first scientist in the picture, as you can see in the slide, Johann Gregor Mendel, who is the pioneer of modern genetics. Around the period of 1886, it took a long time for him to understand the conclusions of his research about heredity. In 1901, the reasons behind the sudden changes were understood due to the mutational theory of Hugo de Vries. In 1902, Walter Sutton observed the paired chromosomes in the cells of grasshopper. Until then, it was not known to anyone. Research started in the direction of finding the nature of genetic material when it was proved that genes are carried via chromosomes, through which 1944 the trio of scientists Oswald Avery, Maclean McCarthy, and Colin McNeil proved that except viruses, all living organisms have DNA as genetic material. In 1961, the French genetics Francois Jacob and Jacques Monod proposed a model for protein synthesis with the help of DNA in bacterial cells. It helped to uncover the genetic codes hidden in DNA. Thereby, the technique of recombinant DNA technology emerged, which has vast scope in the field of genetic engineering. The science of heredity is useful for diagnosis, treatment, and prevention of hereditary disorders, production of hybrid varieties of animals and plants, as we had studied in Standard 9, and in industrial process in which microbes are used. Next, we shall see protein synthesis. Now, before we move on to protein synthesis, we shall study about few terms, as the chapter name is Heredity and Evolution. So, what is heredity? Transfer of characteristics from parents to offspring is called heredity, as we had studied in Standard 9. It is due to heredity that puppies are similar to dogs, squabs are similar to pigeons, and infants are similar to humans. The structure in the nucleus of cells that carries the hereditary characteristic is called the chromosomes, and each chromosome is made up of DNA, which appears dumbbell shaped midway during cell division. Chromosomes are mainly made up of DNA. Now we shall see the structure of DNA. DNA molecule is double helix consisting of two strands. Each strand of the helix is made up of nucleotides. Each nucleotide is made up of phosphoric acid, a deoxyribose, sugar, and a nitrogenous base. Nitrogenous bases are of two types, that's purines and pyrimidines. The purines are of two types, adenine and guanine, and pyrimidines are of two types, that's cytosine and thymine. As we know, the adenine always pairs with thymine with double hydrogen bonds, while cytosine always pairs with guanine with triple hydrogen bond. The helices remains bound due to these hydrogen bonds. There's another nucleic acid named RNA. RNA is a nucleic acid having single strand of ribonucleotides. Each ribonucleotide is made up of ribose sugar, phosphate molecule, and a nitrogenous base. There are four types of nitrogenous base, that's adenine, guanine, cytosine, and uracil. RNA is found both in nucleus as well as in cytoplasm. Now, as our topic is protein synthesis, so according to the functions of RNA is of the following three main types: mRNA, that's messenger RNA, rRNA, that's ribosomal RNA, and tRNA, that is transfer RNA. mRNA carries the information for protein synthesis from genes on DNA chain in nucleus to the ribosomes in the cytoplasm. Therefore, it is called messenger RNA. tRNA, which is present in the cytoplasm, according to the message of the mRNA, it carries the specific amino acid up to the ribosome as per the message coded on the mRNA. The third one is ribosomal RNA or rRNA is the component of the ribosome which helps in protein synthesis. Now we shall see each process of protein synthesis in detail. The first step is wherein synthesis of mRNA as per the nucleotide sequence present on the DNA molecule is called the process of transcription. The nucleotide sequence present in the DNA, as we have seen, is called as gene, and genes control the structure and functioning of cells of the body. Information required for synthesis of protein is stored in genes, and the proteins are synthesized according to the need of the body. Synthesis of proteins by DNA through the RNA is called central dogma. Genetic information is thus used from DNA to RNA, then through RNA for protein synthesis. mRNA is produced according to the sequence of nucleotides on DNA. Now, as you can see in the slide, during transcription, only one of the two strands of DNA is used. The sequence of nucleotides present on DNA strand gets copied in mRNA, that is messenger RNA. Hence, there is always complementary sequence produced on the new mRNA molecule. RNA molecules have uracil instead of thymine present in DNA. That's the difference. Thus, by transcription, mRNA molecule, which is complementary to DNA, is produced. Now, the code for each amino acid consisting of 3 nucleotides is called triplet codon. mRNA formed in nucleus brings the coded message from DNA when it comes in cytoplasm, and this message contains the codes for amino acid. Thousands of triplet codons are present in each mRNA molecule as per the message on mRNA. Amino acid are supplied by the tRNA. Now, as per the codon on mRNA, the tRNA molecule with complementary anticodon is brought near mRNA, and this process is called as translation. Every tRNA brings specific amino acid, and these individual amino acids are joined by peptide bonds with the help of ribosomal RNA, that's RNA. So, as you can see in the slide, the ribosome keeps on moving from one end of mRNA to other end by the distance of one triplet codon. This process is called translocation, and many such polypeptide chains come together to form different and complex proteins. Now, students, as we know that the proteins are essential for controlling various functions of body of living organism. Now you can see the summary of protein synthesis. That's how the RNA is formed from DNA in the nucleus. Then mRNA comes in the cytoplasm where ribosomes are present in the endoplasmic reticulum, and how the protein synthesis or a long protein chains are formed. Sudden change occurring in the gene in the genetic material is known as mutation. Now, due to transmission of parental genes to offspring, there is remarkable similarity between parents and the offspring. But if there is mutation in any nucleotide, then there are changes in the characters of the offspring. Now, as you can see in the slide, in the first picture, you can see that there is a bond, there is a pairing between cytosine and guanine represented by alphabet C and G. But there is a mutation which occurs in the particular code. So in picture 2, you can see that in place of C and G, due to mutation, it is replaced by thymine and adenine. So the sequence is changed. So mutations are of two types. So which, that is minor and major. Minor mutations can also bring about considerable changes, like for example genetic disorders like sickle cell anemia, which is caused due to mutation. Mutation is an everlasting process which leads to the process of evolution, and it also offers proof for Darwin's theory of natural selection, as we will see in the second part of this chapter. Hope so, children, you have clearly understood the process of protein synthesis via 3 steps.